Good morning, everyone. On behalf of the Hepatitis B Foundation and our Hepatitis Delta Connect program, I want to thank everyone for joining today's webinar. My name is Sierra Palecchio, and I am the program coordinator of the Hepatitis Delta Connect at the Hepatitis B Foundation. We're very excited to present today's session on hepatitis delta molecular virology, which is a highly requested webinar topic. And today we have expert speaker Dr. Jean-Michel Pawlotsky, Chief of Biology at the French National Reference Center for Viral Hepatitis B, C, and Delta, and he is also head of the research team, Pathophysiology and Therapy of Chronic Viral Hepatitis and Related Cancers, just to name a few of his credentials. And we thank him for his expertise and willingness to join us on the webinar today. So before we get started, I'd like to review a few housekeeping items and let you know how you can participate in today's webinar. Please note that all attendees are in listen-only mode and will be muted during the presentation. There is also a phone call-in option, and the numbers are listed on the screen. For our international attendees, please be aware that calling in via phone may incur international phone charges. For most people, computer audio is sufficient. And if at any time you have a question during the webinar, feel free to submit it in the chat box on the side control panel, and we'll be reviewing these as they come in, and we will hold a short 15-minute Q&A period at the conclusion of the presentation. And finally, today's webinar is being recorded, and you will receive an email with a link to view a recording of today's presentation a few hours after the webinar completes. So now I'd like to introduce our presenter today, Dr. Pawlotsky. And with that, I'll turn it over to Dr. Pawlotsky to begin the presentation.
Thank you very much, Sierra, and hello everybody. Good morning or good afternoon. It's afternoon here in Paris, and I'm very happy to be here with you today to discuss hepatitis delta virus. So I have no conflicts of interest to disclose regarding hepatitis delta and this presentation. And to start, I would like to remind you that hepatitis delta virus was discovered in 1977 by Professor Mario Rizzetto from Torino, Italy, and his group. Professor Rizzetto identified a new antigen antibody complex they call the delta complex in liver biopsies from patients with severe acute hepatitis. They initially thought that was a new marker of hepatitis B virus infection, but soon realized it was not the case. They used delta antigen to screen sera from patients with chronic hepatitis, demonstrated transmission of infection from seropositive patients to chimps in the presence of HBV, both co- and superinfection, and finally raised the hypothesis that HDV could be a satellite virus of HBV, and that was the beginning of the story. Let's start with a few words of HDV epidemiology. The prevalence of HDV infection is currently estimated to approximately 15 to 20 million individuals. The prevalence varies geographically in HBs antigen positive individuals. It's evaluated to be approximately 5% overall on average, 3 to 8% in the United States. But as you can see on this map here, the prevalence is highly heterogeneous across different areas in patients who are HBs AG positive, with very high prevalence in Russia, for instance, in the former Soviet Union republics, in Mongolia, very high prevalence as well in the Amazon basin, which is not shown here, and slightly lower prevalence in North America or in Europe. And this slide shows you a more precise distribution of the prevalence of HDV infection in Europe. And as you can see, even in Europe, which is geographically relatively small, there is a considerable diversity with countries with a relatively low prevalence, less than 1% in HBs AG carriers, such as, for instance, Finland. My country, France, has a higher prevalence between 1 and 5%, and Eastern Europe has a very high prevalence in HBs AG carriers, 10 to 20% in Russia, and the highest prevalence, above 20%, has been reported in chronic HBV carriers in Romania and in Eastern Turkey. HDV is a variable virus. HDV evolution results from mutation, editing, and recently shown recombination. And this resulted in the diversification of different genotypes and subgenotypes. And at the National Reference Center on Viral Hepatitis B, C, and Delta, we recently published a phylogenetic analysis of 2,100 HDV sequences originating from all parts of the world that confirmed the fact that there are currently 8 HDV genotypes. HDV genotype 1 is by far the most frequent, followed by HDV genotype 5, at least in our collection. And there are many other strains originating from different parts of the world infected with other genotypes, overall 1 to 8. And most importantly, these genotypes are subclassified into different subgenotypes. This is a case here on the left for genotype 1 subgenotypes. You can see at the top the different genotypes, and at the bottom the 4 subgenotypes of genotype 1, the different strains from our collection belonging to these different genotypes. It's full-length sequence analysis, genotype 1A to 1D. And on the right, you can see here genotype 1 and the different subtypes of the other genotypes, non-1, 2 to 8. And as you can see, several of these genotypes have at least 2, sometimes more, subgenotypes identified. This slide now shows you from the same paper the geographical distribution of the different HDV genotypes. And as you can see here, HDV genotype 1 is the most prevalent worldwide, and it's present in most regions of the world: North America, South America, Europe, Middle East, Africa, and Asia. And there are pockets of infections with other genotypes. Genotype 2A is present in China and Northern Asia. There's a pocket of HDV genotype 2B in Siberia, and genotype 4 is present in Japan and Korea. Genotype 3 is highly prevalent in the Amazon basin. And the other genotypes, what we call the African genotypes, 5, 6, 7, and 8, are present in Western and Central Africa, respectively. So a heterogeneous distribution of the genotypes, but genotype 1, and especially subgenotype 1D, is by far the most frequent worldwide. What about the HDV virology? HDV is a very special virus, and this slide shows you the structure of the HDV Virions, which is made of 2 different parts. The first part is the HDV ribonucleoprotein, which is approximately 19 nanometers in diameter and contains the HDV RNA molecule and 2 forms of the HDV antigen, the small HD antigen and the large HD antigen. And this ribonucleoprotein is contained into an envelope, which is in fact an empty HBV envelope, 36 nanometers on average in diameter. This empty HBV envelope contains the HDV ribonucleoprotein, does not contain any other HBV component, and expresses the 3 HBV surface proteins at its surface: the small, the medium, and the large protein. This slide now shows you the structure of the HDV RNAs. The HDV genome is approximately 700 nucleotides in length. It is structured as an unbranched rod-like structure due to extensive intramolecular base pairing. There is 1 self-living ribosome, which is identified here on the genomic form as a red square and a green one on the antigenome. The open reading frame is unique and codes for the HD antigen and is present on the antigenome. And translation from messenger RNAs leads to the formation of 2 different proteins that have a big part in common in terms of sequence: the small HD antigen, which is translated from unedited mRNA, and the large HD antigen, which is translated from antigenome editing of the Ember W site by ADAR1. The slide now shows you the structure of the two HDV proteins, and you can see that these two proteins have a large part in common, approximately 195 amino acids. They are subjected to post-translational modification, including several phosphorylations, a methylation, an acetylation, and a farnesylation. And these two proteins bear in common several motifs: an oligomerization domain, a nuclear localization signal, an arginine-rich motif, and this is for the common part. And the large HD antigen has an additional sequence that contains a nuclear export signal. It's better seen on this slide, and you can see that the additional part not only contains the NES, but also contains a farnesylation signal and the assembly domain. The HDV life cycle is relatively simple, and although this slide is a little bit complex, but the complexity is due to the fact that on the same slide you have at the top the HBV life cycle, which we will not discuss today, and at the bottom the HDV life cycle. And in fact, apart from the very early steps, the entry part, these two cycles have nothing in common. HDV, as I said, is using the HBV envelope. It is recognized by heparan sulfate proteoglycans at the surface of the hepatocyte. And then the HBV receptor, NTCP, takes care of entry. The viral particle is uncoated, and the rest of the life cycle, or the second part of the life cycle, is taking place in the nucleus, and especially the replication step is taking place in the nucleus. So as I said, replication of HDV RNA is intranuclear. The antigenomic HDV RNA is synthesized by cellular RNA polymerase Pol2, possibly also by Pol1. There is debate on this in the nucleolus. The genomic HDV RNA and messenger RNAs are synthesized from the antigenome by Pol2 in the nucleoplasm. HDV mRNAs are then exported to the cytoplasm for synthesis of HDHE proteins, and the genomic HDV RNA is directed to the cytoplasm for assembly of ribonucleoproteins by association with HDAG. The mechanism of replication is a double rolling circle mechanism. It is summarized on this slide. It's pretty unique to hepatitis delta virus. You can see here that antigenomic RNAs are synthesized from HDV genomes through a rolling circle mechanism. Multimeric linear antigen and antigenomic RNAs are cleaved into monomers by the antigenomic RNA ribozyme. And then linear antigenomic RNAs are self-ligated to form a circular AgRNA that serves as a template for synthesis of genomic RNAs by Pol2. So it's the very same rolling circle mechanism. ADAR1, as I said before, is an important protein for regulation of the HDV replication. ADAR1 converts adenosine to inosine by deamination on the antigenomic RNA, and this leads to the change of UAG stop codon for the small antigen to the UGG tryptophan codon for the large HG antigen open reading frame. And by doing so, ADAR1 controls the switch between early phase HDV replication and late phase virion assembly. The late steps are not very well known. They use a Golgi apparatus for budding into the HBV empty envelopes and then lead to export of the mature viral particle. However, there are two types of HDV behaviors according to the fact that the cell is also infected with HBV or not infected with HBV. And it's possible for HDV to replicate in a cell that is not infected with HBV. If the cell is HBV infected, this will lead to a productive HDV infection with the production and the release of infectious viral particles. If not, the HDV infection will be abortive, but there will be production of HDV ribonucleoproteins. And an important point that has been recently shown is that HDV RNA can survive in HBV uninfected cells for at least 6 weeks before HDV propagation is rescued upon subsequent HBV infection. Therefore, the cell can be infected afterwards by HBV, and then infection will become productive for hepatitis delta virus. Pathogenesis and natural history. Briefly, the general features: HDV replicates exclusively in the liver. HDV infection is always associated with HBV infection, and this implies viral interference, and most importantly, a dual cause of liver disease that explains that HDV induces the most aggressive form of acute or chronic viral hepatitis. The mechanisms of liver damage are not well known. A direct cytopathic effect of HDV appears to be unlikely, and what appears to be likely is a preponderant immune-mediated liver damage, but again, this is not well understood. What can we say about the HBV interference? Not well understood either. Superinfection of HBV carriers with HBV leads to a suppression of HBV, and it's well known that patients who are infected with both HBV and HDV had low-level HBV replication, sometimes undetectable HBV DNA in peripheral blood. The mechanisms of this interference are poorly understood. It's been suggested that there is direct repression of HBV expression in co-infected cells. In vitro, a small HD antigen has been shown to strongly inhibit HBV mRNA synthesis and stability, and both forms of HD antigen repress HBV enhancers 1 and 2, and they also activate interferon-inducible MXA gene, and altogether, this may explain that at the end, the expression of HBV is limited, but again, these mechanisms remain largely debated. The natural history indicates that there are two different forms of HDV infection: co-infection, which is characterized by the simultaneous acquisition of HBV and HDV. The course of acute hepatitis in that case is similar to HBV monoinfection, and the risk of chronicity is also equivalent to that in HBV monoinfection, far less than 5%. It's currently estimated that with HBV monoinfection, it's probably less than 1% in adult patients. So this rate would not be modified by a co-infection with HDV. In contrast, superinfection, which is characterized by an acute HDV infection in a chronic HBV carrier, is associated with a standardized relative risk of acute liver failure, which is higher than in patients who are monoinfected with HBV. It's been estimated to 2.4% with a 95% CI between 1.5 and 4.8. And most importantly, chronic HDV infection occurs in the vast majority of superinfected patients, and these patients will have an accelerated course of liver disease with more severe complication. Indeed, if we look at the risk of cirrhosis, this risk has been estimated to be threefold higher in patients who are HBV infected, chronically infected, both viruses, than in patients who have chronic HBV monoinfection. This risk is estimated to be approximately 15% within 1 to 2 years of infection, of dual infection, and 70 to 80% within 5 to 10 years of dual infection, which is obviously a very, very high risk of cirrhosis. The risk of decompensation of cirrhosis is also twofold higher than in patients who have chronic HBV monoinfection, with a probability of survival in cirrhotics of 49% at 5 years and only 40% at 10 years. An increase in the rate of hepatocellular carcinoma as compared to HBV monoinfection is controversial. It's not been demonstrated that the risk of HCC is higher. However, because of the risk of cirrhosis and the risk of decompensation, which are both higher, the risk of death is twofold higher in patients with dual infection than in patients with HBV monoinfection. And only 84% of patients have an event-free survival when they're cirrhotics at 5 years. And this number drops to 59% only at 10 years of dual infection. Diagnosis and monitoring of HDV infection. This is problematic. It's been problematic for a while because there were no standardized assays, and therefore this diagnosis was difficult, especially as far as HDV RNA monitoring was concerned. There are in fact 4 markers of HDV infection: the HDV antigen, total anti-HDV antibodies, anti-HDV IgM, and HDV RNA, which can be detected and quantified. And this slide now summarizes the kinetics of these different markers in patients with an HDV-HBV co-infection on the left, and in patients with an HDV superinfection on the right. In the case of co-infection that does not lead to chronic infection, the presence of HD antigen and of HDV RNA is very transient, a few days in general, very few weeks, and the presence of anti-HDV IgM, as you can see here in the pink line, is also very transient. The IgGs are present for a little bit more time, but they decrease as well and progressively disappear in the vast majority of cases. In the case of HDV superinfection that leads to chronic HDV infection, HD antigen may persist, as you can see here, as well as HDV RNA. HDV RNA always persists, HDV antigen not always, and it's sometimes at low levels that are undetectable. Anti-HDV IgM may also persist, and obviously total anti-HDV antibodies do persist and are extremely useful to diagnose the infection. So at the end, and practically currently, we use mostly total anti-HDV antibody detection by means of enzyme immunoassays and HDV RNA detection and quantification by means of real-time PCR assays to diagnose and monitor HDV infection. The performance of these assays, however, is variable and has been debated, and I would like to show you some results of work that have been performed by the Delta Laboratory under the guidance of Emmanuel Gordien, the National Reference Center. The first study is an HDV Serology Quality Control. There were 4 samples. These 4 samples had positive total anti-HDV antibodies and weak to high positive IgM, and there were 2 negative, and there was 1 negative control, sample D. And as you can see, the 24 labs that tested total antibody detection performed very well, 100% concordance with what was expected. It was not that good for the lab that were looking for anti-HDV IgM, the 14 labs that were looking for them. There were 2 patients with a weak IgM signal, and you can see that concordance was seen only in 43% of labs for sample A and 64% of labs for sample C. So there are issues with IgM detection, and again, I think IgM detection is no longer an important marker. The important marker of HDV presence is HDV RNA in patients who have total anti-HDV antibodies detectable in peripheral blood. So this is now a study also performed at the National Reference Center on HDV RNA Quality Control. That was the first international quality control for HDV RNA quantification, 2 panels. Panel A included 18 samples infected with one of the 8 genotypes, and you can see on the phylogenetic tree on the right the different samples tested, analyzed with other sequences from the reference center's database. And panel B had 2 negative samples and also 2 dilutions of the WHO HDV RNA standard in triplicate. There were 28 participating laboratories, 22 used an in-house assay, 6 used a commercial assay. They were located in different areas: Europe, North America, Australia, Africa, Asia, and Middle East. You can see here the results. The median HDV RNA levels obtained with panel A expressed in log copies per ml on the left and log IU per ml, which has been used to the rest of the study on the right. And you can see that there is considerable dispersion of the results, which goes across approximately 3 logs. And then the detail. The different participating labs were classified into 4 clusters according to their performance. Cluster 1, which is shown here in black, had 8 to 10 samples that were found negative, which is a lot, and the others were largely underestimated. These labs were disqualified. Cluster 2 was not great either. At least 1 sample was missed, and the others were all underestimated. The best cluster was cluster 3, with no sample missed and HDV RNA levels close to the median value. And finally, cluster 4 had no sample missed but the HDV RNA levels were globally overestimated as compared to the reference method. This slide is a little bit busy, but this is showing an explanation for these poor performance of a large number of participating labs. The slide shows the primers and probes used by the 28 participating labs and the number of possible mismatches with the HDV RNA genome target of the 18 positive strains of panel A in the study. And you can see that except for a few labs that were all classified in clusters 3 or 4, all of the others had mismatches in either the primers or probes used for detection, and this obviously explained the poor performance of these laboratories. So what about commercial HDV RNA assay? This slide shows the results of another study performed in the National Reference Center, which tested 3 commercial HDV RNA assays. In that case, it was the light mix assay from Roche, the DiPro assay, and the RoboScreen assay. And the slide shows a blend-outband comparison of genotype 1 HDV RNA levels obtained with 3 different commercial assays using the National Reference Center, the French National Reference Center assay as a comparison. This assay is an in-house assay, which has been established after many years of work based on all the sequences I've shown before from the 8 genotypes, and has equal performance for all HDV genotypes, including the rare African genotypes. So this one was used as a reference and compared with the others. And the blend-outband representation shows you the dispersion of values between 2 assays that are compared, every single commercial assay versus the National Reference Center one. And when the 2 bars are narrow, it means that there is very good concordance between the assays. So here, only genotype 1 strains were tested, and quantification was done on patients coming from Europe and Asia for the upper panel, and from Africa from the lower panel. And you can see here that the 3 assays performed reasonably well for genotype 1 samples, and the correspondence with the National Reference Center assay was pretty good. Now, if we focus on the other genotypes, the rare genotypes, and here you have a representation of genotypes 5, 6, 7, and 8, you can see that the performance of the commercial assays was not as good as the one from the National Reference Center assay, which is shown in each figure on the left. And if we take, for instance, genotype 5, the viral load as measured by the RoboScreen assay were slightly lower than with the National Reference Center assay, approximately 1 log. A little bit lower again for the Roche assay, and most importantly, the DiPro assay was unable to detect them, and not any of them was detected by this assay. This was the case also for genotype 6 on the right, and this was the case for genotype 7, detected with DiPro, but very low level, mostly undetected by the Roche assay, and same as well for the genotype 8 samples. And this clearly indicates that it's not because an assay is commercialized and eventually received a CE mark that this assay has a good performance, and this performance must be evaluated, especially because in our areas, many patients are coming from foreign countries, and a substantial number of them may be infected with rare genotypes which are not genotype 1. So is there any commercial assay that can be confidently used in practice, in clinical practice, to diagnose and monitor HDV infection? Yeah, there is one. This is a French assay. It's not because it's French that it's good. It happens to be good and French. This assay is the EuroBioplex HDV kit. And again, there was a study performed in the National Reference Center on more than 600 samples, most of them infected with genotypes 1 to 8, and negative samples as well. And you can see here the concordance between the results of the EuroBioplex HDV kit and the same samples tested with the National Reference Center assay. 389 were positive in both assays, 198 were negative in both assays, and there were just a few discrepancies. 40 samples that were negative for the National Reference Center assay and positive for EuroBioplex, and 9 that were negative for EuroBioplex and positive with the National Reference Center assay. This slide shows you the relationship between HDV RNA levels measured by both assays. Eurobioplex on the x-axis and National Reference Center assay on the y-axis, and you see the positive and very significant correlation here. And these are individual patient kinetics, patients treated with pegylated interferon or receiving anti-HBV drugs. And HDV RNA levels were measured with the National Reference Center assay in blue and the Eurobioplex assay in red. And as you can see, there was a very good match between the two assays in the values and the kinetics, the individual patient kinetics in these experiments. So what about current HDV therapy? Current HDV treatment is extremely limited, and currently the only drug that can be used in patients with chronic HDV infection is pegylated interferon alpha. The EASL Guidelines 2017 say that pegylated interferon alpha for at least 48 weeks is the current treatment of choice in HDV co-infected patients who have compensated liver disease. Obviously, interferon is contraindicated in patients who have decompensated liver disease, who cannot be treated for HDV right now. In HBV co-infected patients who have ongoing HBV DNA replication, nucleoside analogs or nucleotide analog therapy should be considered, and PEG interferon treatment can be continued until week 48, irrespective of on-treatment response pattern, if it's well tolerated. These results are based on a few studies. I've shown you here one study performed by Heiner Wiedemeyer and our German colleagues in Germany, where patients received PEG interferon alpha 2A plus adefovir in blue, or PEG interferon alpha 2A plus placebo in yellow, or adefovir alone in red. And as you can see, at week 48, end of treatment, a bit more than 20% of patients had undetectable HDV RNA with PEG interferon alpha 2A, with or without adefovir. And this rate at 72 weeks and a follow-up post-treatment was a little bit higher. It was between 25 and 30%. And the figure on the right shows the kinetics of HDV RNA levels with very significant inhibition of viral replication in patients receiving PEG interferon alpha 2A with or without adefovir, and no effect of adefovir alone on HDV RNA replication. So approximately 20 to 25% of patients who respond, unfortunately, these patients do not all respond on the long term. This slide is a follow-up study of the previous one, in which patients were followed for several months after the end of PEG interferon treatment. And as you can see, these 25% of patients who responded initially split into two groups, approximately half-half, patients who have a long-term virological response with still undetectable HDV RNA at the end of follow-up, and patients who experienced late relapses. And this obviously draws back the rate of response approximately 20 to 15%, which is a very low off-treatment response rate for patients with HDV RNA replication. So very poor results of treatment, and these results obviously emphasize the need for new therapeutic options. Research is not as active for hepatitis delta virus than it has been for HCV, and it is currently for hepatitis B. But there are, however, some investigational therapeutic options currently in clinical development. They are summarized here. There are essentially three different approaches that are being explored. Entry inhibitors with one drug called Myrcludex-B, farnesyl transferase inhibitors with one drug called lonafarnib, and nucleic acid polymers with one drug called REP2139. Immunological approaches have also been suggested to be potentially interesting: toll-like receptor agonists, checkpoint inhibitors, but not many studies shown so far. And obviously, HBV vaccination remains the best treatment to prevent HDV infection, but is not providing any solution for patients who are already infected. So to finish, I would like to show you some early data with the three drugs currently in clinical development: Myrcludex, lonafarnib, and REP2139. Myrcludex-B is an entry inhibitor. It's a myristoylated lipopeptide comprising 47 amino acids of the pre-S1 domain of the large HBV surface protein. Myrcludex is administered subcutaneously, and you can see here some early results in UPA mice repopulated with Tupaya melangerie hepatocytes, and that were later infected with woodchuck HBV. And as you can see on the left figure, the administration of the drug prevented HBV replication and made HBV DNA levels undetectable on treatment, whereas in the control groups there was high-level HBV replication. And on the right, you can see also inhibition of HBC antigen production by Myrcludex. So Myrcludex was initially developed for HBV treatment, was not really efficient in, did not have any effect on HBV replication, was not very efficient on HBsAg production, but happened to yield very interesting and very exciting results for hepatitis delta. And this is a study that was presented by Heiner Wiedemeyer at the American Liver Meeting last year. And this study was highly commended because it's full of hope. You can see here patients who received 3 different doses of Myrcludex, 2 milligrams, 5 milligram, and 10 milligram. They were all on long-standing tenofovir for their HBV infection. And you can see that when administration of Myrcludex started, the HDV RNA level went down dramatically, and overall, after a few weeks of treatment, the HDV RNA level had dropped by approximately 3 logs on average, which is extremely spectacular if you consider that we have no other treatment than pegylated interferon alpha. In another study presented by Heiner, you can see here the post-treatment, I think it was in the same study actually, the post-treatment follow-up in patients who received a dose of 10 milligram, and you could see several patients. One patient in red here had a sharp decline of viral replication, became undetectable, and remained undetectable off treatment. All of the other patients had a sharp decline on treatment, but relapsed after treatment, and the HDV RNA level went back up to higher levels. So this is obviously a very interesting option, and we're looking forward for other studies, and we know that other studies are ongoing. The second compound I wanted to discuss is lanifranib. Lanifranib is a farnesyl transferase inhibitor. It inhibits prenylation of the last 4 amino acids of the large Hg antigen, that's CXXQ motif. And this abolishes HDV assembly and secretion in vitro and in mouse models. This drug has the great advantage of being orally administered, but the great dose-limiting disadvantage of yielding GI toxicity, and this obviously limits the use of high doses of lanifranib for clinical practice. This slide summarizes 2 studies. On the left, the NIH proof of concept study with 2 different doses of lanifranib, 100 milligram BID, which yielded a mean log decline of HDV RNA replication of 0.7. And the log decline with 200 milligram BID was minus 1.6 logs. And on the right, the lower HDV1 trial. This is a trial performed in Turkey. I want to insist on the fact that these trials included a very low number of patients, very small number, 4 patients, 6 patients in each group in the first study, 3 patients in each group in the second study. So should be considered very preliminary. 100 milligram TID minus 1.2 log, 200 milligram BID minus 1.6, 300 milligram BID minus 2 logs. And because of the dose-limiting GI effect, it was tested, lanifranib was tested at a dose of 100 milligram BID together with ritonavir. And ritonavir boosting was shown to increase lanifranib concentration by 4 to 5-fold. And as you can see here, with a better tolerance, but again, it's only 3 patients, the viral load reduction was 2.4 log here. And on the right, you can see 100 milligram BID together with PEG interferon alpha. And if we compare to the same dose of lanifranib without PEG interferon alpha, there is some additive effect with a global overall effect of minus 1.8 log reduction. So this is what we have. And this other presentation made by Heiner Wiedemeyer at the EASL meeting this year. It's not 2017, I think it's 2018. This is a lower HDV4 trial. It's a phase 2 open label rapid dose escalation study of multiple twice daily doses of lenacapavir boosted by ritonavir. And you see the effect of the compound on viral replication on treatment with different effects due to the use of different doses. But most importantly, you have some post-treatment follow-up here. And as you can see in virtually all cases but one here in orange, this case had the kind of blip after treatment and then became undetectable. All of the other cases remained HDV RNA positive, and the viral load went back to the initial values in the vast majority of these patients. So a transient effect on treatment, but this effect is not maintained after the end of treatment. The third compound I wanted to discuss is a nucleic acid polymer, Rep2139. This is a phosphorothioated oligonucleotide, which binds with high affinity to amphipathic alpha helices in protein structures. The mechanism of action in HDV infection is very much unclear. It's been suggested that this compound could inhibit viral entry, but not very solid data to support this. And also it's been suggested that there would be a blockade of HBsAg release and secretion of subviral HBV particles. This remains to be confirmed. The compound is administered by weekly 1 to 2 hour infusions, and obviously this mode of administration is also a big issue because we can't think about people using lifelong treatments administered by 1 or 2 hour infusions. Results were shown with this study. This is a paper published in the Lancet Gastroenterology and Hepatology last year showing the administration of Rep2139 initially with the infusion inducing a decline in serum HDV RNA levels on the left. The problem is that the study is a little bit polluted by the early addition of PEG interferon alpha, which further enhances the decrease of HDV RNA replication. And during follow-up, you can see here that one patient remained with undetectable HDV RNA, and the other patients for which follow-up is available had relapses post-treatment after Rep2139 and PEG interferon treatment. And on the right, you can see in the same patient the HBsAg levels, and interestingly, the patient who apparently cleared HDV RNA also cleared HBsAg, and the other patients had modest to sharp declines of HBsAg levels, and the vast majority of those who had a post-treatment follow-up relapsed after the end of treatment administration. So this is where we are, and this brings me to my summary slash conclusion. So overall, there are approximately 15 to 20 million individuals who are infected with HDV worldwide. This virus has a high genetic diversity. There are 8 genotypes with different geographical distributions and a number of subgenotypes. And the virus also has a very complex life cycle with a particularity of using host cellular proteins and especially enzymes, making it very difficult to target specific viral enzymes and making specific drug development more difficult than it used to be for HIV, HBV, or hepatitis C virus. HDV infection induces a most severe form of viral hepatitis. Diagnosis and monitoring are based on total anti-HD antibody detection and HDV RNA quantification. There are very limited therapeutic options. Currently, only pegylated interferon could be used, and it could be used only in patients who have compensated disease. And there is active ongoing research on new therapeutic options. However, the results may be considered very preliminary, and we're really looking forward to seeing more and more results with these 3 new therapeutic options. Finally, I would like to thank the members of the National Reference Laboratory in charge of the Delta Laboratory, especially Emmanuel Gordien, Frédéric Legal, and Ségolène Brichler, who have, I think, yielded a very important contribution, especially in the evaluation of diagnostic tools and the establishment of the genotypes, subgenotypes, et cetera, et cetera. So on this, I would like to thank you very much for your attention and can take questions if any. Thank you very much.
Thank you so much, Dr. Pawlotsky. So now we will begin our Q&A portion of the webinar. So please submit your questions via the chat box. And while we wait for more of those to roll in, I'll start with our first question. Someone is asking, is the Europlex HDV kit available in the US?
I don't think so. It is available in France and in Europe, and the reason is that it is easier to get the CE mark than to get the FDA approval. It's a small company. I'm not sure they have all the resources to get through the FDA approval and do these huge clinical trials you have to do to get the FDA approval. And this is a pity. It's a pity because there are other markers that are not available in the US for that reason. I'm thinking about the HCV core antigen, or I'm not sure quantification of HBsAg is available in the US actually for the same reason. So unfortunately, not this assay is not available in the US so far.
Thank you. Another question is, would a longer treatment with pegylated interferon alpha be a better option for patients with Hep B and D, longer than 48 weeks?
Yeah, I definitely think so. There have been a few small-size studies with longer treatment. As long as peg interferon is well tolerated, I think going up to 2 years is possible, and with probably slightly better response rate. Probably get up to 40% initially, but the late relapses may happen as well. But if treatment is well tolerated, it's worth trying. Yeah.
Great, thank you. Another question is, are most HDV patients genotyped, and is there a commercial test for this?
Though they are not genotyped in practice because this has no implications for treatment and care in these patients. The reason why it's important to know the genotype is to understand that some rare genotypes will not be detected or quantified by the assay. So you don't need to genotype, but you need to know exactly what is the genotype distribution in your area to know whether or not you can rely on the assays that are used for diagnosis. If you live in a place where it's been well demonstrated that 100% of your patients are genotype 1 infected, it's not a big issue. If you live in Europe, in North America, in places with heavy immigration from countries that have a high endemicity for these rare genotypes, it's important to know the genotype distribution in your area just to know whether the assays you're using are reliable and whether or not you're missing patients who are positive and in fact come negative from the lab you're working with. And this is something we had to face in France because we have many immigrants from North Africa and Sub-Saharan Africa living in the country, many of whom are HBV infected, and a substantial proportion have HDV infection. And it became very obvious early that these patients were not diagnosed by the genotype 1 based assays.
Great, thank you. Another question is, if hepatitis delta replication causes a suppression of hepatitis B, why is co-infection more dangerous?
Because co-infection is not related to, because the liver damage is not related to the level of replication, because neither virus is cytopathic. What is aggressive for the liver is the immune response directed to both viruses, and therefore there is an interferon that reduces HBV replication, but the immune response is against two viruses, and it's therefore more aggressive. There's more inflammation and more rapid progression of liver disease.
Right, thank you. So another question, how do you differentiate an acute HDV infection from a chronic one?
Well, it's difficult. Chronic HDV infection generally occurs in the context of chronic HBV infection. So the patient is chronic HBV carrier. The co-infection is easy because the patient has acute hepatitis B, so you have anti-HBc IgM. And you diagnose acute hepatitis B if at the same time you have HDV RNA and anti-HDV antibodies. It is an acute co-infection with HDV and HBV.
Thank you. So if hepatitis delta can live for 6 weeks without hepatitis B, what are the implications of this? And is there a scenario that this might present in a patient?
I think it's a way by which the virus established its persistence in cells. This means that if HDV production is not sufficient in HBV infected cells, the virus can wait for the cell to be infected to be produced. It's just a way by which the virus is dependent on HBV, but not too dependent, and can infect more cells than the cells that are HBV infected. And this may be a way by which the virus adapted to the fact that it reduces HBV replication, on the other hand. So this is, balance based on Darwinian evolution, just hypothesis. But this is a way by which a virus can persist. It's the best way the virus found to persist in cells in the liver.
Right. So another question. Someone is wondering about, could there be multiple infections along with hepatitis B and D? Is it common to see other co-infections or tri-infections?
You mean infection with other viruses, or
Yeah.
multiple infections with different genotypes or strains of HDV?
I think they're referring to possible other viral infections like hepatitis C or HIV.
Yeah, there have been many patients initially at the beginning of the HIV epidemics, especially the intravenous drug users, the people who inject drugs, were infected with the 4 viruses. And we've seen in the early years of the HIV epidemics, HIV, HBV, HCV, HDV infected patients. To be very honest, most of them died, and we see much less right now because most of these patients just died. They had very advanced liver disease, and even when they were treated for HIV, their liver disease was out of control. There was no treatment for hepatitis C at the time except peg interferon. They had B plus C plus D, very severe liver diseases, and many of them died from the liver. So it's possible, and there are still high-risk groups of patients who have the 4 viruses together. Yeah.
Right. So another question. For patients who respond to treatment and do not relapse, do they lose HDV antigen or do they just remain RNA suppressed?
They remain RNA suppressed. They generally don't have HDV antigen detectable. HDV antigen is very transiently detectable, and in chronic carriers, it's rarely found also. So in a patient who would have a sustained virological response, there would be no detectable HDV antigen anyway, but you would not test for it. So that's the point, yeah. And they keep the anti-HDV antibodies, but what we don't know is whether or not these antibodies will disappear in patients who have a sustained virological response. There are not enough patients and not enough follow-up for these patients so far.
So are the implications of that they may be able to become infected again?
It's a very good question. This I don't know. I know it for hepatitis C, where there is no protection against reinfection, superinfections. You can be infected with different strains. And this is a reason why I asked you what the question was initially, because I don't know if multiple infection with different HDV strains have been reported. I will add the question to Emmanuel Gordien, who probably knows that.
Yeah, that is a complicated question.
Yeah. I'm not sure there are data actually, but it's an interesting question.
Yeah, that seems to be the problem. There's not always data there's just hypothesis at this point for a lot of delta. So yeah. We'll do one more question. Someone's asking, how can you manage hepatitis D after a transplant? Or what happened? And I'm not sure if that's within your realm of
No, I mean, these are very difficult patients. You obviously try to prevent HBV reinfection, but Delta is still there, and there is potential reinfection with Delta. Yeah, but there are not that many patients that would be ideal to have the oral drugs available. But as you've seen, what is in development is not that promising. It is exciting, it's interesting, but it's not that promising as an oral treatment which would fit with post-transplant administration. There are issues also with interactions, drug-drug interactions. PEG interferon could be used post liver transplantation, but it's difficult, it's complicated. It's been used at the time of HCV treatment post-transplant with some results, but it's not ideal. It's difficult. It's a difficult question. It's a difficult situation.
Right. So it's definitely hard to determine the best kind of endpoint or how to really deal with Delta, even in cases of transplant. It might not solve everything.
Yeah, it's difficult in the simple case, so it's hyper difficult in the complicated case of post-liver transplantation.
Mm-hmm. Well, thank you for your answers and for delving into those difficult questions. That's all the time we have today for questions, everyone. But I want to thank Dr. Pawlotsky again. And Dr. Pawlotsky, could we finish off with just a few key ending points from you before we sign off today?
Well, I think they were on my summary slide. So 15 to 20 million people infected. It's a lot. It's not negligible. The virus is complicated. It's not easy to have a great idea waking up in the morning and say, oh, I will target this enzyme and we'll have a good drug. It's much more difficult than that. So I think there are two things that need to be done: raising awareness on the frequency, severity of the disease and the unmet need, and stimulating basic research to find options, new targets, and develop new drug for these patients, because it remains a real unmet need, and everybody's focusing on other successes, which is fair enough, but there are 15 to 20 million people with HDV infection worldwide.
Thank you so much, Dr. Pawlotsky, and thank you for your insight and expertise. It's much appreciated.
My pleasure.
All right, absolutely. All right, everyone. So I just want to remind you to please fill out the post-webinar survey and let us know if you'd like to opt in to receive more information from our program. And please do let us know any future webinar topics that would be of interest. And also remember to check out our website and follow us on social media. And again, thank you all for participating today in the webinar and for joining, and have a great rest of the day. Thank you again.